Okay. So now we talk about the principal component analysis. It's another way of doing kind of clustering analysis and unsupervised learning. If you think about a very simple scenario, supposedly if out of all the genes on this microarray, you can only pick one gene to separate the samples. You don't really care whether it's separating cancer or normal, but you just try to capture as much of the differences of all the samples as possible. What would you pick? Would you pick a gene that doesn't vary a lot among all the samples, or you will pick a gene that varies a lot among all the samples? You should go with the latter one, right? You pick you pick a gene that is very, very different among the different samples. So now, supposedly we give you a chance to pick a second dimension. The second dimension is you pick another gene, second gene. Would you also want to pick another gene that has big variability among all the samples? Yes, but then do you want to pick that gene that's correlated with the first gene or completely different from the first gene? Completely different, right? And so the completely different is called orthogonal, which means they are not correlated. Okay, so imagine we have to do this for many, like for every additional thing you can, additional dimension, you can pick something. You will just pick the genes that have good variability but different from the rest. So principal component analysis, instead of each dimension only pick a single gene, in each dimension it picks all of the genes but just give them a different weight. Okay? Yeah, so it performs a linear transformation of the original data, which means you consider all the genes but give them a different weight. And so at the end you can just get that measure. And so PCA, in some sense, will give you kind of decompose the original data into different dimensions, and in each dimension, using a linear combination of all the genes to capture the maximum variation in all the samples. And then in the second dimension, it's going to try to give another weight to all the genes so that it will capture the remaining variations in all of the samples. But it's an orthogonal weight from the first weight you give in the first dimension, so that they are kind of not correlated with each other. Yeah, so when you do PCA, all the components are decomposed simultaneously. So let's look at a very simple situation here. Supposedly you have a teapot. The teapot is a three-dimensional thing, but supposedly we only want to give it two-dimension. Let's not worry about the lighting. Imagine this is just, white versus black. And you can look at the teapot in many different angles. You can project it from the top, from the side, from this way or this way, right? And you will see or even tilted, and you will see the teapot in different ways. I would imagine the dimension with the least information will probably be this, because by the way, you will not see the sprout coming out. It will just be black, right? The whole thing being black. You don't really see enough variation. You will not know that this is the teapot probably. But this dimension is probably pretty good, right? Even from two dimension, you can clearly tell this is a teapot. But if you are given this, you're like, is this an urn or a teapot? You're not so sure. But if you look from the top, let's not worry about these things. Yeah, that's pretty good, right? You will get some idea. So you can see there are many possible ways to do this projection. The nice thing about principal component analysis is that it automatically identifies the projection. So the first principal component will give you that projection that kind of give you the best information, all the genes together. Okay. So what does it do is, so this is your original data, and this basically in each of your original gene. Remember, each of in a microarray experiment, this could be 20,000 genes here. But if we give the first gene some weight, the second gene some weight, and third gene, so every gene is given some weight. The X is your original data, and each of the genes is given a weight. You just multiply their kind of the weight and add all of them together. Now you summarize all of them into a single number. And so now the first dimension for this first sample will be just represented by a single number. And PCA can automatically identify the correct weight so that when you look at this, you can separate all the samples in the best way. At this point, we don't really know which one is cancer, which one is normal. We just want to make sure that we have a good spread of all the different samples along the first dimension. Okay? And the weight that you give each gene is a column vector, and this is normalized, so the sum of square of all the weights will be 1. Okay, so it's kind of a unit vector. And it just makes sure that if we project all of those samples, you will have the best separation of, maximize the variance of the projection of the observations on the Y variable. Based on just this first Y, you will have the best separation of all the samples. Okay, so that's the first principal component. So you can imagine if we have a 2-dimension spread. If you project in this way, you have this. You don't really have a very good separation. The best separation would be this way. And you can see the samples are really quite spread out. You capture most of the variations. And so here, we will just be giving X and Y each a weight. Or you can imagine this, if this is X1 and this is X2, each are given a weight to come up with this single dimensionality of, in this equation, it will be the Y, right? In this case, and you can separate out the samples. Yeah, so the direction of, you can imagine the weight that you give to each gene is the angle you picked to look at the teapot. Because if it's a unit vector, you can imagine in a 3D-dimensional space, the X, Y, Z, that the coefficient you give will determine what the projection direction, right? And so then you kind of convert the original higher dimension into a single value, right? So the direction is given by the eigenvector, which corresponds to the largest eigenvalue of the matrix. Yeah, the second vector is another direction, which is orthogonal, which is uncorrelated with the first one. And you are hoping that this will capture the second highest variance, which comes to be the eigenvector corresponding to the second highest or second eigenvalue. Okay? Yeah, so, and this is achieved through a process called a singular value decomposition. And so if you have your original data and you do singular value decomposition, it will convert the original data into 3 kind of components, or not components, they are 3 things. X is your original data. U is the relative projection of all the points. So U is, is how much you get from the first principal component. What is the value you get for the second component? So every sample will have a score. And then from all the components, you will get their kind of projection level. V is the projection direction in here, which is, in this case, it will be the coefficient that you give to each of the genes. It's the weight that you give to each gene, right? So V is the unit vector. The direction of the first projection is V1. The direction of the second projection is V2, and so on. So V1 corresponding to the highest eigenvector with the largest eigenvalue. So it's the linear combination, which is the relative importance of each different gene on this microarray in order to really separate out the samples. So you can imagine if we do first principal component, the gene that has really, really large variance in the first principal component will be given a pretty high weight. The second gene, if it has also very high variation, but also quite different from the first gene, will also be given a high variance, right? Things like that. So this is the first principal component. So you imagine this is two-dimensional data. The first dimension, you try to capture the variation in this way. But the second principal component will be orthogonal to the first one that really capture the remaining variation in the data, which is smaller than the first one. In the high dimensional data, you will also do the same thing. Every dimension, it will capture less and less variations, but each of them also is a linear combination of all the genes. Okay, and you can look at the top, say, 10 dimensions or 20 dimensions and so on. And by the way, so the D is a scaling factor, which is the eigenvalue of your total matrix. So remember in here, V1 is a unit vector. We only give it the direction. We don't really capture the size of the variations we are capturing. And so the D is really the scaling factor. And if you look at this matrix, it's diagonal matrix, just have values on the diagonal. And from the first number, it's bigger. The second one is smaller. The third one is smaller and smaller and smaller. And they basically tell you how much to scale each of the principal components. And the squared of this D matrix, the values, measure the variations that are captured by the top number of principal components. So for example, if you have eigenvalue of this, so basically it tells you how much variation the first principal component is able to capture. In this case, the total data has huge variation. If you just use a linear combination to project all the genes already, how much variation is it able to capture? And if you sum up all the Ds, that's the total variation. Each of this divided by the sum is how much this particular principal component is able to capture on that particular principal component. Okay, yeah, so that's why the first principal component captured the biggest variation. The second principal component captured the second largest variation. The third will capture the third largest variance, and so on. Okay, and so you can imagine, sorry, it should be, yeah, so U1, D1 is the distance along the V direction from the original. This is the actual kind of, how much the original thing is projected using this directionality. Yeah, so is the expression value projected onto the V1. Is your kind of, remember original Y is this thing. Using this projection on each of the gene, previously if you just do this projection, you will get a Y, but if you multiply also by the D1, that's the actual projection because you also have the scaling factor. Okay, so V2 is the second projecting direction, which is orthogonal to the principal component 1. And the V2, D2 is the second principal component and capture the second largest variation of your total data. Okay, so that's principal component. Let's look at an example. So supposedly, we are able to get the expression data of some blood. This is gene expression of some blood from many different individuals. And they are considered, when they came in, there's like healthy individual, some risk of getting a cardiovascular disease. And this is some people who already have some abnormality or dysfunction of their heart. And the last group is this patient already having a heart failure, which is probably already going to the ER, right? So if you get their blood and do gene expression, and then you run the principal component, basically each of the samples, we will weight their genes so that we can capture the biggest variations of the samples. Then we plot them on this graph. Now it's a 3-dimension or first the three principal components, right? So the first principal component are here, the second is going this way, the third is going this way. And then after we have projected into the three-dimensionality or three principal components, we can now look at the sample labels to say which ones are totally healthy, which one has some little risk, which one has some already abnormalities, and which one is kind of going into the ER. And you can see, okay, the samples actually cluster together, which is a good thing. And this group is, yeah, this is healthy. You can already see the sample separation. And principal component analysis you can also use as a way for dimension reduction and clustering. And you can imagine if you have a new sample coming in, you can also put it in this clustering and see whether it's cancer or normal, or in this case, whether it's a healthy individual or they are going to the ER. Okay? Yeah, so supposedly you have a new sample here. You can use this original projection on the 3D to give you some predictions. Okay? The nice thing about principal component analysis is, remember, k-nearest neighbor is kind of a black box. You just like, oh, whatever look like other things. You don't understand why. However, we can look at the weight of the variables in each component. So supposedly this is the first principal component, and these are the weights that you give to each of the genes. So based on the weight, you can tell, as we mentioned, the first the genes that have very big variations will have a better weight, whereas the genes that don't really vary much, they will have very tiny weight, right? So you can see, so in this case, X1 and maybe X3, they are more important than X2 and X4 in the first principal component. And it can actually offer you some biological insight. You know, what is it giving you? You can even imagine if you rank all of the genes based on their first principal component, you can run the gene set enrichment analysis and see what that first dimension is. You know, maybe you will pull out, this is about some pathway being perturbed. The other principal component is about something else. So imagine if you have a disease versus normal case, you look at all the separations, yeah, maybe it will tell you, okay, the reason this person got this disease is because one pathway and another pathway or something else went wrong, right? So you can capture that from the principal component analysis. So PCA and MDS, multidimensional scaling, are both good at dimension reduction, but they are kind of different in that MDS just try to preserve the pairwise distance. But PCA want to make sure that the first, like all the top principal components, just try to capture as much of the variability among all the samples as possible. So they try to optimize a slightly different objective function. Yeah, so PCA is also a good clustering method. It can be conducted on genes. You can look at whether genes cluster. So it's just you transform the matrix, or you can also cluster on the samples. So in this case, we are clustering on the samples, and that's kind of the sample classification question. But you can also use it to classify the genes to see whether the genes have similar behavior in different samples. So PCA is only powerful if the biological question is related to highest variations in the data set. As we mentioned, you know, like we're hoping that the top three principal components can already separate your cancer normal. And then you have a chance of predicting whether a new sample is cancer or normal. There are cases when the top component is not really of the biological question you are interested in. Can you imagine what that situation is? Yes. Sorry? Oh, ethnicity. Okay, yeah. So in this case, it's right. It's not because of the disease. It's because of some other confounding factors, right? But confounding factors can mean a lot of things. For example, batch effect is another confounding factor, right? So it can be a very, very good way to identify batch effect as well. So supposedly, this is a published study. They look at the expression microarray data. If you don't normalize it, just look at it, the index. I think these are samples coming from different batches. say you are trying to do cancer normal or some comparison, this is not for, sorry, this may not even be a classification question. Just supposedly you process a lot of lung cancer samples. And ideally, you want to look at the samples that can separate the cancer subtype. But the reality is, if you don't normalize, it's crazy. It's like not really very good. But if you do quantile normalization using RMA, okay, they remove some of that, but you can see now these are just samples from different batches. Okay, remember we mentioned quantile normalization just do array by array separation. If there are condition-specific variations, that's kind of a treatment, right? So RMA will not remove those kind of batch effect. And so if you can draw a PCA plot after you finish quantile normalization and RMA, and the samples really cluster by batches. Yeah, so in homework 2, we ask you guys to look at the samples using clustering, but you can also use PCA, and you can see that there is clear batch effect. And then after you use combat to normalize the data, now at least we know that the samples coming from different hospitals or sample processed in different months are together. So if you have this, okay, that's good. And then you can ask, is the first principal component really separating, say, a subtype of cancer versus another subtype of cancer? Okay, yeah, so you can use PCA to not only identify the batch effect, potentially you can also use PCA to remove the batch effect. Can you imagine how you do that? Exactly. Remember, this is the original data, right? If you have the singular value decomposition, you can estimate, how much weight do I give? Basically, each of the gene is given a weight. Supposedly, this is the second principal component, or maybe the first principal component, which is completely confounded by the month this experiment is done, or the hospital this experiment is done. Then you estimate that the parameter for that principal component, and you remove this effect from the original gene. That essentially can also remove the batch effect from the original data, right? So the first gene, instead of the original value, you multiply by a little bit of something and you remove it out. And similar to kind of combat, you give it a different, you remove a little effect from each gene, right? It's like a linear model of removing some treatment effect. And that treatment happened to be a batch, right? Questions? I think that's all about principal component analysis. Yeah. Yes. You are not zeroing out. Remember, originally for one sample, right? For each sample, you have the expression of gene 1, expression of gene 2, gene 3, gene. But if all the samples belonging to the first batch have huge batch effect, then basically you give this gene a weight and kind of remove that effect from the original expression matrix. Right? Each gene, you will remove it by the corresponding effect. You take it out. Okay? Then
in the SVD, like the how would that you do that there?
I think you can do this. Yeah, not the SVD just give you the matrix, and you say, okay, I remove the first out, and the remaining one is the remaining one. You will still capture the the remaining biggest variation, but at least you remove the one major variation, which is coming from the batch. Okay, yeah, Ali. You can, yeah. Well, yeah, sometimes you can do that. So, for example, you first see, this is the month, and that one is the hospital, and the third one is really the cancer normal. Yeah, you could do that. Yes, another question. Can you speak up, please? No, you can completely remove it. Yeah, because each gene has a weight, right? You just, the original gene is subtracted by a little bit of that effect out. Okay? All right. Oh, did I start recording? Ah, shoot, I think